Good afternoon, everyone, and I would like to thank the organizer for giving us this opportunity to present our work. My name is Afsar Ahmed, and I'm the Chief Scientific Officer for Aeterna Health. And my work is focused on the novel autologous immune cell therapies. I'm just going to give you a brief outlines. What is the autologous immune cell therapies? When you're talking about any kind of therapy, there's only mainly kind of two types of therapy. One is that non-living therapy, another is that living therapy. Non-living therapy are the small molecule drugs, peptides, and as well as like antibodies. When I'm coming to the live therapy, which is called the living cells that we use for the therapy. And in live therapy, there's two types of therapy. One is that non-immune cell as well as the immune cell, and our work is focused on the immune cell therapies. And what is actually meant here, the autologous immune cell therapy, is this, that those immune cells that we produce, which is going back to the same host. This is why it is called the autologous immune cell therapies. So I'm just going to start with this global market of the immune cell therapy as well as its rising trend, just to understand the current its demand as well as the interest in this actual field. So this is the cancer therapy market, which is currently one of the largest in medical therapy, where it is expected to grow by 8.9% over the next 10 years. And within this cancer therapy, there is another one called cancer immunotherapy that pretty much changed the circumstances of this cancer immunotherapy over the last 10 years, which is also expected to grow at the same rate over the next 10 years. But in the cancer immunotherapy, the light therapy, which is currently being dominated by CAR-T, which is currently expected to grow almost like 3 times than this previous one. Even this market value is quite low, but it is expected to grow much more higher. And because the immune cell therapy, which is an emerging concept, which is its full potential, it remains to be found. And there is like a growing interest in this therapy. For example, CAR-T initially started in 2012. It has only 12 clinical trials, but now it has more than like 1,200. Having said that, that's the immune cell therapy, which is also associated with painful side effect, and majority of them are not quite yet successful against the solid tumor. And because of this, there is a constant need for the better, efficient therapeutic targets, and because of that, we are actually working towards it. So I'd like to start with some basic concept about the immune cell therapy, which is started with the interactions of different type of immune cell with the cancer cell. And the way this immune cell interact with different cancer, like a cancer cell, that varies between one type of immune cell to other. They have a different way of interacting this kind of like a cancer cell. And based on this concept, different kind of immune cells therapy is currently in the market. For example, like a tumor infiltrating lymphocytes, lymphocyte activated killer cell called the LAC cell, cytokine induced killer cell, cascade prime induced cells, which is called the CAPRI, natural killer cells therapy, as well as like a recently been used over the last 10 years, genetically modified T cell, which is all the TCR the cell, as well as like a CAR T cell. These two are the currently that genetically modified cells. But what you can see here is this majority of this therapy, which is has either some drawbacks or some kind of like limitations. For example, some of this, like a TIL therapy, is a very low abundance, which is not quite efficient to deliver the therapeutic outcome. Some of them has some toxicity because of the IL-2. This needs to be infused with these cells, and that is called the toxicity. Other is also causing some cytotoxicity. Even for the CAR-T, the severity is actually so high, it can also cause some death. And out of actually all these like therapies, cascade prime, the CAPRI cells stands out quite different because it's not only have any toxicity, but it is also used in a combined effect of different kind of immune cells. Based on these limitations, there are some immune cell therapies is currently suffering a number of drawbacks, and majority of them are here, been highlighted here. For example, it has a long manufacturing process. For CAR-T, it goes up to like 4 months. Some of them induce IL-2 and that is also causing some toxicity. Majority of the cell therapy which is currently exists in the market, they also have a low to severe side effect. And low abundance of the effective immune cell within the body is also another like a drawbacks. And immune cell are also once which has been used for the therapeutic purpose, they also lose their function over actually time, and some of them not even live longer. And perhaps one of the most significant limitation of this current immune cell therapy is the solid tumor, because so far the immune cell therapy is not been so successful against the solid tumor. As you can see, structure that tumor microenvironment is a very complex structure. It's not only have this tumor cell, but also have some other associated connective tissue as well as other supportive cell. And the last but not the least is also saying that tumor cells can also undergo a lot of changes. It can change its antigen that can also prevent it from being eradicated by immune cells. So in order to address the current limitations in the immune cell therapy, what actually happens, like we started our work on the Capri cell therapy, and I'd like to highlight here a few of the strength of the Capri cell therapy. Capri cell therapy is, to our knowledge, is the only therapy exists today which use both innate immunity as well as like adaptive immune cells, and it is using the patient's cancer information is stored in the circulating monocyte to differentiate naive T cell into actually effective T cell. So in this way, it is maintaining the natural interconnectedness of different immune cell within actually the body. And instead of using like a one type of immune cell, it is using a combination of immune cell and other effector cell within the Capri are NKT, B cell as well as like a dendritic cell. So it is a one way of elevating the natural way of immunity within our body. And it does not require any immunogenic peptide or genetic manipulation, and therefore it is physiologically friendly. It does not cause any side effect, and this is the only therapy exists which does not cause any side effect within the immune cell therapy class. And it can also be delivered within a very short span of time, like 2 weeks, which has a very quick manufacturing process. And Capri cells, even though in this talk I'll be focusing on the cancer only, but Capri cells have the wider implication. It can be used not only for cancer, but also for autoimmune disease, infection, as well as like a boosting general immunity. In order to understand that what are the cells been involved in Capri, as I said, that this is not just a one type of immune cell, it has a different type of immune cells. So we analyze the Capri cells, and it is majority of them, more than 85% of the Capri cells are CD3 T cell. For that reason, it can be considered as a T cell therapy. Within this CD3, even though it is mainly dominated by CD4 and CD8, it also has Tregs and Th17 cells. Apart from the T cells, other remaining effector cells of Capri are dendritic cell, NKT, as well as like a natural killers, like a killer cells. The main reason we are here in this expo, because in Aeterna Health, we invented a new version of Capri, which is we call that I cells. And when we tested these I cells against a number of cancer cell along with I cell as well as like a Capri, what excited us is this: I cell has much more potent anti-cancer phenotype than Capri cells, which means it is actually give us a green signal that this therapy can give us the right optimum therapeutic outcome, which is currently immune cell therapy market is actually lacking. So once we get this outcome, we wanted to move forward and wanted to find out the more details, the differences, how these I cells are different from the currently existed Capri. We looked into the immune profiles. And once we looked into the immune profiles, we found that within this Capri cell population, I cell has a much higher proportion of B cell compared to the original Capri. And it is quite interesting to know that there is a current interest going on, a lot of actually research going on actually the B cell, because B cell was initially thought as an antibody producing cell, but it is also playing a very important role to maintaining the innate immune cells as well as like a cancer immunotherapy. So in our eye cell, it is showing that the proportion of the B cell is higher compared to original Capri. And another cells, we found the difference between the Capri as well as like our eye cell is that the Th17 cells. Even though Th17 cells is pro-inflammatory cells, which has playing a very important role in cancer, but there is a kind of like a literature there which is showing that there is a controversy about the role of this Th17 cell. Based on the circumstances, it can be acting as a both cancer promoting as well as like a cancer inhibiting cell. But in our cases, we found that the Th17 cell lower number is better for better therapeutic outcome. And for the immune cell to prevent cancer, one of the critical factor it needs is the pro-inflammatory cytokine like TNF alpha, IL, as well as like interferon gamma, and these are the pro-inflammatory cytokine that can prevent the cancer cell instead of like a supporting cell. For that reason, we wanted to see that our eye cell has this like a pro-inflammatory marker or not. When you looked at it, that the levels of circulating levels of pro-inflammatory marker released from these cells, we found that both the levels of TNF alpha as well as interferon gamma are highly elevated in our eye cell compared to the Capri, and it makes perfect sense that this is why our eye cells is much potent cancer killing activity compared to original Capri. And perhaps the most exciting findings for our work is this: we found that central memory T cells are highly elevated in our eye cells compared to actually Capri. And the main reason it is so exciting for us because the T cell differentiation is a very critical factor that can cause the dysfunction in most of the T cell therapeutic treatment, because when T cell get differentiated, it loses its persistence, it also loses its proliferation activity, it also loses its functions. But on the other hand, central memory T cell that maintain their proliferation potential, it can also highly persistent, they also remain functional. So having the difference between central memory T cell as well as terminally differentiated T cell, they are the major driving force to determine whether this immunotherapeutic will be successful or not. And this is why it is quite exciting news for us that we found that eye cell contains majority of this central memory T cells number has got elevated in our eye cell compared to the Capri, and that gives us an indication that why our eye cell therapy is much more efficient compared to Capri. And lastly, this is also one of the critical part of our work is this, that HLA cancer cells express number of surface protein, and that can help the immune cell to recognize them and to actually to be killed. But you may have a very fantastic immune cell therapy, but cancer cell, they're very smart. Sometimes they lose their ability. Sometimes they hide their expression of this HLA peptide to escape from the immune cell therapy. So we wanted to see what happened to this cancer cell when they are treated with both Capri as well as like eye cell. So when here we use only HT20 to 29 cell, which is one of the most difficult cell to kill, which belongs to the colorectal cancer group. And when these cells were treated with Capri, we found that their HLA-E expression is elevated. And HLA-E is quite important for the colorectal cancer because it is high expression is associated with favorable outcome. But when you side by side, when you treated these cells with the eye cells, we found that HLA-E expression much more elevated in eye cell compared to Capri. And this is also showing here the protein level here that HLA-E expression in these cells much more elevated when they're treated with iCell compared to Capri, which is also showing another indication that our therapy, which is making the cancer cell expressing those kind of proteins which can be easily recognized by the immune cell as well as to be killed by the immune cells. And this is just the highlights of our current program overview. In Aeterna Health, we do the biobanking where we process the immune cells and then we isolate the immune cell and then we do the liquid biopsy as well as preserve these cells. And our second part of the work that is currently working on the clinical trials where we expand those cells, and the technology I just explained, that technology can be used and changed according to the clinical needs. We can use these cells for cancer patient as well as for immune use as well as for autoimmune disease. And hopefully the main reason we are here, hopefully we should be able to one day to expand our techniques outside Australia and hopefully in China as well in the future. And this is the brief outlines of our current pipelines, that we are not only focusing on cancer, we are also focusing on autoimmune disease, anti-aging, as well as like immune boosting. Anti-aging is quite exciting news for us because anti-aging is something that most of the aging process is started with the gradual dysfunction of our immune cell, and we believe that our technique can help prevent the anti-aging process. And this is just a future trend of the immune cell. Instead of the majority of the trial, which is going on one type of immune cell, the current trend been using the combination of different type of immune cell. So this is why our iCell technology fits well into the current trend of this immune cell therapy for into the future. So I'd like to finish in here. And if you have any questions, and I'll be happy to answer any question for you. And thank you for your attention.